Hi guys, it's Rob from Royal Balls. With all the fuss about DG recently, I thought I'd do another video, so let's kick off by having a look at my Desert Ghost clutch from Christmas last year. And of course everyone will remember my Christmas Day Desert Ghost clutch, and nothing has changed since the genetic revelations. This was a visual to visual Desert Ghost pairing, 5 babies, all of them visuals. This is the one remaining male, so I think this is leopard yellow belly. You can see the belly there has the typical leopard zippered belly, and we are now starting to see some flames coming up the side and a little bit of that typical yellow belly leopard interference in the pattern here, so a slightly unusual pattern. So I am thinking that this is yellow belly leopard Desert Ghost, and this is a male, and you can contrast that with the yellow belly Desert Ghost female. She has much more blushing up the sides and also the typical yellow belly head stamp. So she's a carbon copy of mum. This is a female, and obviously that's a holdback. This male is not a holdback. So leopard yellow belly, or just leopard Desert Ghost, and this one yellow belly Desert Ghost. Another holdback female from that Desert Ghost clutch, and this is Lemon Blast. Again, another obviously visual Desert Ghost, super clean Lemon Blast Desert Ghost. And this girl has been a little bit slow to eat. She's not the best eater and a little bit slower growing, but she is growing and she is eating. So another holdback. This is Lemon Blast Desert Ghost. There we go. So I got 2 females and 3 males, and I'm not keeping any of the males because I do already have a visual Desert Ghost male. And the pick of the clutch, here's one for Samantha. Pastel Leopard Desert Ghost. This is the nicest looking snake. In person it is just phenomenal. So this is Pastel Leopard Desert Ghost, and it's a male, and just look at that awesome pattern and those awesome colours. For me, this is the nicest snake of that bunch, the one that glows the most, and obviously pastel in Desert Ghost just works. Awesome snake, Pastel Leopard Desert Ghost. What we're going to do today is have Mr. DNA show us how to convert the pipe cleaner demonstration into a Punnett square using just simple notation. The first example is a very simple mono hybrid Punnett square using Enchi paired to an Enchi. Let's take a look. Alright guys, everybody has seen the Punnett square and pipe cleaners in action, so we're going to create a Punnett square today. We're going to look at pairing an Enchi to an Enchi. Now again, the male is in black and the female is in pink, and I think everybody is used to seeing a modified gene which represents Enchi. We are going to put it on the locus where Enchi resides on the genetic code for the male, and because it's only one copy of Enchi, this is going to be a visual Enchi, but it is actually the heterozygous condition. One side of this double helix of DNA contains the modified gene and one side does not, and the same for the female. At the locus that Enchi resides, we are going to put a copy of the modified gene, and on the other side of the locus, the double strand of DNA, we have no genetic modification. So we're going to pair an Enchi to an Enchi. We split the genetic code in half for both the male and the female because we only need half the genetic code, and we're going to recombine it with half the genetic code from the female. So in my previous videos you will now know that what we have done is made a haploid cell from a diploid animal. This is the original condition with a double strand of DNA, and when we make the gametes, in this case sperm, we are creating haploid cells, and these haploid cells 50% of the time will carry Enchi and 50% of the time will not. So let's have a look at the various permutations that we could get from pairing an Enchi to an Enchi. We could pair this side of the male's DNA, the haploid that contains the genetic modification, and we could match it to this side of the female, and we get 2 copies of Enchi. So we'll put that outcome in this square, 2 copies of Enchi. Now this same haploid sperm cell carrying the genetic mutation for Enchi could also meet a haploid egg cell from the female which is not carrying the genetic mutation, and in this case we only get 1 copy of Enchi, and we'll put that here. Now we take the other half of the male's DNA, and these are haploid cells which are not carrying the genetic mutation for Enchi, and we pair that to this haploid egg cell from the female which is carrying a copy of Enchi, and we get 1 copy of Enchi. Let's take the same haploid sperm cell not carrying the mutation, and this time it meets an egg cell from the female which is also not carrying the genetic mutation for Enchi, and that goes in this square here. There are no genetic mutations from that particular pairing. So only 4 outcomes, and we've demonstrated how to do that using our pipe cleaners. So here is our super Enchi with 2 copies of Enchi. Here are 2 single copies of Enchi, and here is 1 offspring that doesn't carry any of the Enchi gene. Now it's important to realize that what we are doing here with our pipe cleaners is creating the haploid sperm or haploid egg cells, and for the male there are 2 potential outcomes for the haploid sperm cells. We can have sperm cells which carry 1 copy of the genetic mutation, or we can have sperm cells that do not. So we can actually write on our Punnett square, rather than use our pipe cleaners, we're going to use this as the label for the gametes for the male. So one of his gametes may carry a copy of Enchi, and one of his gametes does not carry a copy of Enchi, and to differentiate the 2 we use capital E for the gene which is carrying the genetic mutation, and we use small e to represent the non-modified gene. So here are the 2 potential outcomes that we can have for the male. We can have one side carrying the genetic mutation, the other side not. And on this side here we can do the same for the female. So the female can provide eggs which carry a copy of the genetic mutation or do not carry a copy of the genetic mutation. So here we can construct a Punnett square now without the help of the pipe cleaners. So all we do is look down and across like noughts and crosses. So this box here is E, E. That is our super Enchi. This box here is E, E. That's our Enchi. This box here is E, E. That's our Enchi. And this last box here is E, E. So we have 2. Hello, Pepper. Yes, I know. So we have 2 small e's on this particular locus. Both genes are not carrying the modification for Enchi, so this is a normal. So rather than use the counters, the coloured counters and the pipe cleaners, we can actually construct a Punnett square using just the letters and this notation. So here is our monohybrid 4-square Punnett square, and we've just used it in the case of an enchi to an enchi, which is written big E little e to a big E little e animal. We've calculated what the gametes would be, and we can transfer those to the top row and the side column, big E little e and big E little e, as the potential gametes from both parents. So now we can start to fill in the matrix for each of the squares. We take big E from the top and big E from the side and produce an EE, or homozygous enchi animal. Keep going, filling in all the squares. And in the top left we have big E, which is our super enchi. We have two squares which are big E, little e, and these are both enchis. And we have one square which carries no genetic mutations for enchi, little e, and this would be a normal. Okay, so now we're going to crack on and use this principle for our dihybrid desert ghost scenario. If I do this right, it will be easy. If I do it wrong, it won't. Okay, so what I want to get across here is that DG has two different genes involved that reside on two different chromosomes, which means we are dealing with two different loci with two pairs of alleles. DG does not exist as DGA, DGB. It exists as DGAA and DGBB. We need to have a copy of DGA and a copy of DGB in order to express as a visual. DGA by itself, even in homozygous form, AA will not work. DGB by itself, even in homozygous form, BB will not work. So in this example, heterozygous DGA, heterozygous DGB, the locus for DGA can have no mutations, written small a, small a. It can have a mutation on one side, written big A, small a, and it can have a mutation on both sides, written big A, big A. In this case, we're looking at heterozygous DGA, written big A, little a. Exactly the same is true for the locus for DGB. It can exist with no genetic mutations, which is written small b, small b. It can exist with one genetic mutation on one side of the locus, written big B, small b. And it can exist with two copies of the mutation on both sides of the locus, written big B, big B. In this case, we're looking at het DGB, which is written big B, small b. So I asked Mr. DNA to demonstrate for us how to use a simple 4-square Punnett square and just 4 pipe cleaners to demonstrate a dihybrid Punnett square, and he's agreed to do that. So you don't have to be worried that this is going to be complicated. Alright, so now that we've constructed a monohybrid Punnett square using 4 squares, we're going to move to the Desert Ghost dihybrid. Now, it's more than likely that Desert Ghost is more than just 2 separate genes. They've already identified a third, possibly a fourth, and maybe even more. But based on the information that we're given to date, the two most important genes are what they're calling DGA and DGB. So two separate chromosomes, two separate loci, two separate pairs of alleles. It's important to realise with the Punnett square that the labels on the top and down the side are the potential gametes from each parent, and once we get that concept into our head, it becomes very easy to fill in the matrix regardless of how many squares there are. So let's hand over to Mr. DNA, and he's going to show you how to use the 4-square Punnett square to define what the potential gamete or sperm egg cells would be, using the example of a het DGA het DGB to het DGA het DGB. Let's have a look at what that looks like. Okay, so traditionally we've used our 4-square Punnett square on monohybrid, a single gene, and we've computed all the different permutations that we can get for that monohybrid cross. And in the case that I've just shown you, we've used an enchi to an enchi, and I've shown you that we can actually label each of these rows and columns with the potential combinations of gametes that each of those snakes could produce. Now let's look at a dihybrid mutation like DG. This is going to be incomplete. It's more than likely that DG is actually a trihybrid or a quad hybrid mutation, but for now we're going to deal with just the dihybrid DGA and DGB mutations. So I'm going to show you how we can use our 4-square, very simple Punnett square and just 4 pipe cleaners to determine what the genetic combinations are that a male or a female DG could produce depending upon her genetic sequencing. So let's get rid of the female. We only need one sex. Here is our male, and we are accustomed to splitting the male's genetic material and using only one half or the other half to combine with the female. Well, there are actually 18 homologous pairs of chromosomes within that male's DNA. 18 homologous pairs, and we are going to look at just 2 of them. We are going to look at the homologous chromosome that contains the locus where DGA resides, and we're going to look at the chromosome, and it's a different chromosome. It is not the same chromosome. We're looking at 2 different loci on 2 different chromosomes with 2 different pairs of alleles. So we're going to look at the chromosome where DGA resides, and we're going to make this male heterozygous for DGA. On this chromosome he carries 1 copy of DGA, and on the other side he's not carrying the genetic mutation. Now let's move to the chromosome where DGB resides. So I'm skipping out the intervening chromosomes, and remember there are 18 pairs of them. We are going to make this chromosome where DGB lives, we are going to make this heterozygous for DGB. So when the male makes his sperm, every single one of those 18 pairs of chromosomes divides to produce a haploid sperm. So let's do that. We're splitting up this chromosome and this chromosome. Our haploid cells can actually exist with DGA and DGB in 4 different combinations. So let's look at what those combinations are. We can combine this side of this chromosome with this side of this chromosome, and we get no genetic mutations in this square. We can combine this side of the chromosome with this side of the chromosome where DGB lives, and you will see that we get 1 copy of DGB. Let's put that there. Now Let's take this copy of this chromosome and pair it with this chromosome when everything lines up to produce the sperm cells, and there are 18 chromosomes all doing this. This one has one copy of A and no copies of B. So let's put that there. We can take this copy of this side of the chromosome and pair it to this chromosome here, and this time we get one copy of A and one copy of B. So for a male, that is heterozygous for DGA and is heterozygous for DGB, and that would be written A B B. Okay, so on each chromosome this male is carrying one copy of the genetic mutation for DGA and one side is not. This chromosome is carrying one copy of the genetic mutation for B, written as capital B, and this side is not. So for this particular male, what are the genetic combinations in his sperm? Well, he can produce sperm which have none of the mutations, which would be small a, small b. This square here has one copy of the genetic mutation for DGB, so this would be big B and little a. This has one copy of the genetic mutation for A, but no copies of B. This side has A and B. One copy of A and one copy of B genetic mutations. So we've used the same 4 Punnett square, but this time what we've used it for, instead of the final outcome, we've used it to define the configuration of the sperm cells from this particular male, who is heterozygous A and heterozygous B. So he can produce sperm which carry a copy of the genetic mutation for both A and B. He can produce sperm which carry a copy of A. He can produce sperm which carry a copy of B, and he can produce sperm cells which carry no copies of either of the genes. 4 potential outcomes for generating sperm from this male. When we do with the same with the female, you can see that we have 4 potential outcomes from the male, we have 4 potential outcomes from the female, so when we combine the 4 potential outcomes, we have a total of 4 times 4, which is 16. Let me show you the Punnett square for that. So here is our 16 square dihybrid Punnett square, and on the top you'll see I've put the label het DGA het DGB to a het DGA het DGB, and this is written A small a, B small b, to another animal which is A small a, B small b. We've just used our 4 square Punnett square to work out what the potential gamete combinations are for each parent, and they go on the top row for one parent and down the side for the other parent. We know that big A can exist with big B, big A can exist with small b, small a can exist with big B, and small a can exist with small b. So we label the rows and columns with the potential gametes from each parent, father on the top, mother on the side, to create a matrix. So we fill in each of the squares according to the labels in the rows and columns, and in this particular case we have AB from the top, AB from the side, so this becomes homozygous A, homozygous B, or AABB. Let's fill in all 16 squares, and I'll leave this up for a moment so that you can actually study it, but if you want to look more closely you can always pause the video. So which of these combinations are going to be visual DGs? We know that we need at least one copy of A and one copy of B to be a visual, but the best ones are homozygous for one and heterozygous for the other, or homozygous for both. So 5 out of 16 of these animals have the potential to be really nice looking DGs, and 5 out of 16 is slightly better than 1 in 4, or slightly better than a typical het-to-het pairing. We do have 1 out of 16 animals in the top left hand corner which is homozygous for both A and B, which is your 1 in 16 for a typical double recessive. However, there are an additional 4 animals which are carrying heterozygous A and heterozygous B, which also have the potential to be visual DGs, and that is 9 out of 16 animals have the potential to be visuals, which is not a typical het-to-het result. 7 out of the 16 animals do not carry the necessary genetic sequence to be visual, and that includes animals which are homozygous A but carry no B, and homozygous B but carry no A. So these ratios are neither typical double recessive or simple recessive, which is exactly what you would expect, because DG is not. It's polygenic. So here I've listed out for you in words on the right hand side all the different combinations that we could produce that have the potential to be visual DGs, produced from a het DGA het DGB to het DGA het DGB. It is not the matrix you would expect from a simple recessive het-to-het pairing. However, when you look at it in its basic statistical terms, it's very close, and that is probably why we haven't noticed until now that DG was actually polygenic. Alright guys, I hope you were able to follow that. So now that we've filled in a matrix for a 16 square dihybrid Punnett square, we can have a look at some other examples. We can have a look at a homozygous DGA homozygous DGB to a homozygous DGA het DGB, and have a look at what the outcomes for that are, and they will be totally as expected. So here's the matrix for homozygous DGA homozygous DGB to homozygous DGA het DGB, and here half the babies are going to be homozygous for both, and half of them are going to be homozygous A and het DGB. Next we'll look at a homozygous DGA with no DGB to a homozygous DGB with no DGA, and I've seen many times on social media that this is now, based on how a Punnett square works, is going to be the best pairing combination you can make to get the most number of visuals. Incorrect. Let's take a look at that example and let's have a look at why that will not work. This matrix is interesting because I've heard many people say that actually pairing homozygous A with no B to homozygous B with no A is actually the best combination to get the most number of visuals. And you'll see that in this 16 square dihybrid Punnett square, that is not true. You will get 16 double heterozygous A and B animals, but we know that these animals are not in most cases visuals. So if you pair homozygous A no GGB to homozygous B no DGA, your results are going to be mixed and you may be disappointed. So once again, I'm using the spreadsheet published by Kinova to reinforce the point that phenotype and genotype are not necessarily the same thing. We are conflating and confusing the two. Visual DG is a phenotype. It is the look of the animal, and whilst it may imply a certain genotype, the genotype is not guaranteed. I believe that the visual to visual DG pairing that Kinova has done that produced some animals which didn't look like DG, and that's a subjective interpretation, those animals did in fact test genetically homozygous DGA and homozygous DGB. Their genotype is DG, their phenotype is not. Genetically, you can produce desert ghosts from desert ghost to desert ghost pairings that do not look like desert ghosts, but they are desert ghosts. Their genetic makeup makes them genotypically desert ghost. More than likely they're missing an essential ingredient, and this is potentially the third, fourth or even fifth different gene that's involved in this polygenic mutation. Alright guys, once you start to work with Punnett squares and you start to look at all the different scenarios, as I've pointed out in the scenarios that I've modelled, they are actually remarkably similar visual outcomes to what you would expect if Desert Ghost is a simple recessive. Obviously the combinations are more complex and there are more of them, but the ones that actually will generate a visual DG in statistical terms, it's almost the same. Yes, you can choose certain pairings that will not give you visual DGs, and your Punnett square will explain why that occurs. But if you have plenty of visual DGs to work with, a visual DG by definition has all the genes that it needs in order to express as a visual. And if you use those in your pairings, you can't go far wrong. All the correct genes are going to be in the mix somewhere. Likewise, if you pair a visual to a het, you're also going to get the right combination of genes to express as a visual. And you can draw the various Punnett squares and see that you will get, in most cases, at least as many visual DGs as you expect to get. Very rarely will you get less. Het-to-het pairings are a little more problematic, and that affects me because I have a number of double het to double het pairings this coming season that I'm trying to make the double visual Desert Ghost clown. In most cases where you've produced your hets from outstanding quality visuals, they will still contain the necessary genes. Some of the offspring will obviously not be DGs, but there is still the possibility of getting the double visual from that pairing. If I get some surprises, I probably will need to re-look at that, but in most cases it will not change what I am doing in my pairings. Visual to visual will still work, visual to het will still work, and het to het is still a good bet. Only in rare cases will a het to het throw up something unusual, and the dihybrid Punnett square, or even the trihybrid Punnett square when we find out about DGC, will be able to explain that. But in most cases you probably won't even notice. Thanks for watching. Don't forget to share, like and subscribe, and we'll see you on the next one.